Thank you very much. It's such a pleasure being invited to speak here and to meet with so many colleagues, friends, and people who've worked with us together over the years. It's fantastic listening to Dr. Chichana and just seeing how much, progress and how much impact a single person like Dr. Chichana can make on our thinking and the way we approach these problems. I also want to give, an acknowledgement to Safwan Alamari, who's the next speaker, who's currently a fellow working in our laboratory and is actually applying to neurosurgery residencies this coming year. And he's just a superstar, as you'll see when he gives his talk about what he's done, the work with in animal models for the type of goals we're trying to achieve. And he helped me very much for this talk today. I have involvement in several, it's complex to accomplish what we do, and we need to work with companies as well as academic centers and as well as crossing many different disciplines. I think that, we're going to talk about this morning, the importance of tissue is axiomatic to us as neurosurgeons, oncologists, and so on. And it's interesting to go back to, so much of what we think about goes back to Harvey Cushing and to our founders in the field who really did understand the issues. They didn't have the tools that we have today, but they understood the issues. And this is, some quotes from Harvey Cushing in 1930. Firstly, he looked at supravital staining and he said the cells which have not undergone changes due to fixation or cutting were very valuable to look at immediately from the operating room. And then he talks about why is that so important? Well, the indication for the prognosis that can modify the measures accordingly. And so much what Dr. Chichana was talking about today was understanding the individual patient's prognosis, what we're trying to achieve in modifying our surgical approach based on that. And here's Cushing saying the same message with different tools and determining what particular operative procedure to pursue and how radical or otherwise an extirpation of the lesion is desirable. And then the importance that he stressed of an immediate diagnosis so that the surgeon can utilize that information in order to best design best treatment possible for the patient. So the importance of accurate pathology in modern neurosurgery is of course critical. And some of the things we're going to talk about this morning are the evolution of the World Health Organization, classification of CNS tumors, how important that is for clinical trial design and therefore assessing what we're accomplishing, and high quality tissue collection and advancement of the pathology assays is critical now as we reach approach that the pathology is different than what we used to be looking at, and we need to think of it in different ways. The need for intraoperative immediate histopathology and the approaches using machine learning, advanced microscopy, and other technology that we can assess the tissue, and therefore, it's again, Dr. Chichana pointed out, sometimes modify what we're doing and how radical it be and what portions of the tumor are so critical to remove. And then the management plan, prognosis, and precision medicine, and the advances in liquid biopsy, which, as Jim Rutka said, is the mantra of what we're trying to achieve that would advance our field so radically. And we're at very early stages of that, but we need to develop technology to make that more applicable to neurosurgery as it is being applicable to other fields. And then finally, I would add to that the importance of tissue is for researchers, because we are at the beginning of what we can do in terms of understanding biology and developing the therapies. And with proper tissue, cells, stem cells, neurospheres, we can then study the biology and develop much better therapies than we currently offer our patients. So let's look at the pathology and how that's evolved and actually the rate of that evolution. 1979, the Blue Book was published by the World Health Organization on the classification of brain tumors. And that was after many, many years of pathologists arguing and discussing what is the best approach to classifying brain tumors, and became the Bible of our field. and stood really until the second edition in 1993 when immunohistochemistry started to become important, and they reclassified, or they added to the classification, still based on morphology and H&E sections, primarily, but looking with immunohistochemistry, getting more information, and that evolved 1993, then 2000, it was revised 2007. Then when genetic and molecular targets and understanding became prevalent, the fourth edition came out in 2016, and very rapidly thereafter, you can see the rate of change in pathology is increasing as we begin to understand the biology better. And this is becoming critical, of course, for clinical trial design and for prognosis for patients. And, again, as Dr. Cicciotta pointed out, it influences what we do in the operating room as well in terms of how that impacts on prognosis. So the pathology, how does it influence clinical trial design? Well, when we did the studies on gliadel, for example, we really just, people had malignant gliomas. When we did the recurrence, it was recurrent glioma. At the upfront study was malignant glioma, it was grade 3s, grade 4s, every subtype was thrown in. And to show a benefit statistically was actually very challenging because there are many patients in that group that we know by their molecular subtypes are going to be resistant to every therapy that we currently have. And there are also some patients in there that really benefit enormously, but we made a hodgepodge because that's where the field was at in the 1990s. And despite that, some treatments came through and showed a statistically significant improvement, but it was a very hard bar. And many treatments that might have been very beneficial to some patients failed because they couldn't treat that entire big lumped up together group. So as things evolve, we start to look at, biomarkers, MGMT, for example, in the Timidor studies, you could take that group out and see that there was much more of a benefit in one group versus another. And then we're evolving now that we're looking at different molecular markers and then taking smaller and smaller groups and looking at more targeted therapies, which is really, again, one of our goals of finding these biomarkers is does it benefit patients who have those? So it gets harder to accrue patients into large studies. You can't get the kind of numbers that we got when we took all comers. On the other hand, the impact may be much greater because as we subdivide into smaller groups and look at the right therapy for those people, we can decide whether a therapy works or doesn't work. So I think that's the direction that we're going. And of course, ultimately, when we talk about precision medicine and individualized therapy and personalized medicine, that is the direction we need to go. We can't throw the same things at everybody because they have some broad, brain tumor diagnosis, but rather, as we start to understand the biology, we should target the therapies. And we're moving in that direction. We're not there yet. So what is it about the tissue and how critically important it is to get the proper tissue for us as neurosurgeons and for our oncology colleagues and pathologists? And are we doing a good job with that? And are we getting the information that we need? So this is a study from Spain by Castello Domingo. I think this was the first major study looking at RNA sampling from biopsies of various brain tumors. And what they found was somewhat disappointing, but important. And they said that really only 27% of the samples, 27% of the samples, I guess it's not disappointing, it's uplifting because it's only 27% were below the standard measures for RNA integrity. So, if that's important, we were not doing a great job. Now, one of the variables they looked at is how long the tissue was kept, whether it was snap frozen, had a much better outcome than if it was for 15 minutes or 30 minutes. These are all variables that we don't talk about, and we don't have uniform standards of how quickly the tissue is processed, and yet it's a critical difference in terms of preserving it. Also, how many biopsies were done, and whether with multiple biopsies from separate areas, it was more likely or less likely to achieve, useful RNA integrity. And half the patients when there were 3 biopsies done, did have useful, but you can see that a great deal did not. And then multiple sampling is also important because in this slide, in this study, shows that even within each tumor specimen, there's a huge heterogeneity as you study them. So it's important to, in order to understand the biology of the glioblastomas, to not just rely on one sample that says, yes, you have tumor, and then move on and use the cavitron and get rid of the rest of the tumor, but rather to study multiple areas, multiple different imaging sites, and to study the tumors in that way. And in that sense, the NICO device, the automated tissue preservation system, is very valuable, and we've you'll hear more from other speakers about its clinical use, and you'll hear from us about the next talk about its use in animals, but in the biology that we've been able to establish with that. But this system allows rapid, reproducible, preserved, standardized method of obtaining tissue. Now, some of the problems with intraoperative histopathology, this is a work that Masaru Ishii and Gary Galley in our department looked at, that artifacts can lead to incorrect readings. These were neuroblastomas that they took out endoscopically, but showed that you can have artifacts that say that there's no tumor, that it's just necrosis, and then it turns out there is tumor, and the other way around. So again, tissue preservation with the minimum amount of artifacts is critical. The other thing is interesting as we look at health systems, and unfortunately, more and more of my life is working at all the other forces on our field, in addition to just what the challenges of the laboratory and the operating room. But there's actually a lesser number of pathologists available as the workload is increasing. As we demand more of our pathologists, we want immediate answers. We want to know tissue subtyping. There are actually fewer. If you can see here, let me see if I can, that the number of pathologists is actually decreasing. The other thing is that if you look at the US versus Canada, the demand in Canada is pretty steady. And Canada is just a steady state, great place. But in the United States, the demand per pathologist is increasing for how much we expect for them for neuro-oncology, while the number of pathologists is steadily decreasing. So it behooves us to automate what we can to make uniform and utilize the resources in the optimal way. Along those lines, and again, this is Dr. Chechana's work with Dr. Quinones and others on looking at methods of looking in the operating room, intraoperative assessment of glioma surgery using subcellular resolution with multiphoton microscopy. And you can see here the astounding images and how they compare to H&E from their studies. And the platform can identify different regions of the tumor, and they're matched here with the histology. And also that with deep learning and AI, that we can classify in the operating room malignant, non-malignant, and you can see that matching it against the pathologists, that the accuracy is incredible. So newer technologies for looking at the tissue itself. And it can be used in real-time surgery to guide the resection. And I know out of time, Dr. Chechana didn't show that, but these are the type of technologies that they use to get the super marginal resections and to be sure that they're getting tumor out and it's the viable tumor. The other thing is portable intraoperative stimulated Raman histology, another approach to really looking at the tissue in the operating room, comparing it to H&E with remarkable correlation, and using machine learning and coming up with accurate diagnoses, which will help us in terms of multi-institutional studies, multiple institutional care of these patients. It can't just be sort of the elite centers that do this kind of work have a lot of manpower, but really to make it more uniform and more available, and also to make it more consistent among all the centers as that evolves. So, and then another one of our former colleagues and continued deep friend, Alex Olivi, who spent 27 years with us at Hopkins, starting as a fellow in our laboratory, and now is for many years now the chairman at the Catholic University in Rome. And this paper was just awarded like last week by the CNS as the best paper in the tumor area for the upcoming meeting, but using machine learning-based predictions of early recurrence in GBM, a glance towards precision medicine. Again, using these criterion and using machine learning techniques to give better prognosis for patients, and also to guide how we carry out our surgery. Final thing is the goal of using liquid biopsies. Can we establish what's going on with molecular markers and DNA fragments and so on for many cancers, which has been very successful for, but brain tumors, which we've just trying to get there. Chetan Bedagauda in our department has been one of the real leaders in this area. This paper in Science Translational Medicine has now, 33,000 citations. It's the most cited paper that Science Translational Medicine has had. It was the first paper on liquid biopsies. And it's amazing, in terms of, our pride and our specialty, that the first author on the explosive field of liquid biopsies is a neurosurgeon leading that effort. But unfortunately, of the tumors looked at, brain tumors were the least predictive and the least valuable in a sense, because blood-brain barrier and the markers don't get into the bloodstream as well. And here you can see the different, what a liquid biopsy looks at: exosomes circulating, tumor cells, fragments of DNA, and the advantages and disadvantages of each of those different methods. But all of them can be used with both blood and increasingly with CSF as well. And, what's the potential applications for the clinical care for our glioma patients? Well, there are times that we can't establish, or it's harder to establish diagnosis with tissue biopsy, some of the deep-seated lesions, multicentric lesions, people who are not candidates for surgical interventions. It's a very relatively, or maybe totally, safe way of following patients longitudinally, understanding when there's true recurrence versus treatments are necessary. And of course, as these tumors evolve and mutate and change, it's a matter of designing better therapies as we evolve those therapies to gear for that individual patient. And so that's the mantra of what we're trying to achieve with the liquid biopsies, and also to give a more accurate assessment of what the prognosis is for each individual patient. As this is, a group got together to look at liquid biopsies in glioma, the review said that at this time it's not applicable to neuro-oncology, but obviously that we need to get there and we need to change that. So there are several, not enough, but ongoing clinical trials investigating liquid biopsies in glioma patients, and different factors that need to be addressed in the design of that. Now, this shows that the finding looking at all cancers, that, for example, bladder cancers, lung cancers, and so on, have a spectacular rate of being successful with liquid biopsies, but you can see medulloblastomas, gliomas are quite poor. So the shift became, well, maybe instead of looking at blood, we should be looking at CSF, and a study was done, again, led by Chetan Bedagauda, looking at detection of tumor-derived DNA and CSF of patients with primary tumors of the brain and spinal cord. 35 patients were looked at, and at least one mutation was identified in each tumor using targeted or genome-wide sequencing, and patient-specific mutations were then used as biomarkers to follow them longitudinally. detection levels of CSF, tDNA in 74% of patients. All patients that abutted on the CSF, like medulloblastomas, ependymomas, high-grade gliomas that were near a CSF space, were detectable 100% of the 21 patients that were identified in that manner. But no CSF tDNA was detected in patients whose tumors were not directly adjacent to a CSF reservoir. So the question is, how could we improve that? And the study is now underway, and it's at several centers, but we're Shane Betagauda and Graham Woodworth at University of Maryland have R01 funding to be looking at MR-guided focused ultrasound as a way of opening the blood-brain barrier preoperatively, then sampling blood samples, and then seeing if the marker accuracy can be improved, or prevalence can be improved by doing that. So looking at plasma levels of ctDNA, extracellular vesicles, brain-specific protein, and looking at it with and without blood-brain barrier disruption preoperatively, and then of course matching that against the findings intraoperatively. So those studies are underway. They have found that focused ultrasound can increase the sensitivity of liquid biopsy by the amounts shown here for the different markers. And there are several ongoing trials, both at Hopkins, University of Maryland, the MGH, Toronto, and WashU that we know of. I'm sure there are others as well. So where are we with all of this? I mean, accurate pathology plays a key role. I think that's axiomatic. That's not a new discovery for the evolution of our field, and improves the care that we can deliver to our patients. Precision medicine creates a rapidly growing call for high quality tissue collection and precision analysis. And it's critical that we work together with engineers, biologists, other people to meet the need in molecular era. And I would say the other thing is that if we're going to advance our field, we can't just do it with the existing tools, that we need to do better to really move on the explosion and extraordinary increased understanding of the biology of cancers and of tumors. And in order to do that in the laboratory, we really need to have better, more consistent tissue from the operating room. And, I would conclude, we were talking last night about, has the field really changed in the last 50 years? And I would say that there are phases that we've gone through. And firstly, my answer would be enormously. But the first was, Cushing's time, the original pioneers. It was all about, could surgery be done? Could a patient survive an operation? Could you do, the arguments, could you do total resections versus partial debulking? So what was feasible? But the idea that surgery was safe was many, many years in evolving. Then the next revolution, which occurred in the 1970s and early 80s, was imaging, where we were doing so much guesswork and very crude methodology, skull x-rays, neuroencephalography, really things that are unimaginable today. With the advent of CT and then MR and then tractography and connectomics, now our ability to image details of the brain and use it in a navigation way has been revolutionary and has changed our field and changed the safety and allowed for the kind of things that Dr. Chichana spoke about, that we can truly, not the way Gandhi said he did a complete resection or a hemispherectomy, and in the photographs in his paper, you can see that half the tumor is still there even after a hemispherectomy. But we can accurately know that what are we accomplishing in the operating room. And then the next, I think, really revolution has come from our understanding of immunotherapy, biological markers, beginning to understand the role of stem cells, because maybe we've been missing the target of what we should be shooting for in terms of where our therapies are guided. And that evolution, and the pathologists are leading a lot of that. And of course, the laboratory is leading that. That is dependent on us as neurosurgeons producing representative, accurate, well-preserved, consistent tissue so that it can be studied, and therefore treatments can be designed properly and so that the patients have the proper prognosis and outcomes maximally. So I think that's our next challenge, and I think that we're moving nicely in that direction. Thank you very much for the chance to share some of these thoughts.
Thank you so much, Professor Brem. Open it up for any questions, and while you're considering and getting your microphone, how does all of this and improved tissue collection, how does it change the workflow intraoperatively for the neurosurgeon?
Well, it's always been our job to collect tissue. I think with the NICO system that we're going to hear about in the laboratory and clinic, I think as you systematize it, it actually should require less manpower because instead of, having people that sort of run the tissue immediately and saying, we're doing a study and we have to get someone quickly in operating in multiple operating rooms at night, weekends, multiple rooms going on at once. How do we do all of that? This provides a very systematic, sort of automated approach that says that we can do this without, actually with less expense, because with less people expense and less need to sort of be demanding of all of that. So I think this workflow, the goal is for the workflow to be simplified, but much, much more accurate and consistent.
Dr. Rucker.
Henry, a great keynote. Thank you very much. Question is related to clinical trials that you mentioned, and the importance of those, especially now based on the molecular diagnostics that will likely pave the way towards targeted therapies in the future. But the question is, like, how will we make it equitable for those patients treated in centers that may not be either university-based, non-academic, smaller practice-based, where they don't have biobanking service, where they don't collect these specimens, and these patients, as a result, will not have access to the clinical trials? That's first question. Second question relates to your comment about needing more neuropathologists. It's interesting. You mentioned, however, machine learning. If I was a neuropathologist, I'd be kind of worried that I wouldn't have a job about 5 years from now, because everything's going to be automated and there'll be less need to look at the actual histology. Maybe a different type of neuropathologist will be needed, who will understand all the molecular aspects, but less in the way of formal, histopathology.
Well, to answer the first, and my wife is here, who's doing automated learning on breast imaging and radiology, and the fear that radiologists have that they may not be necessary. I think the reality is just the opposite, that the more information we get and the more we use the automated information and computer systems, the more we need human integration of what it means and how it's put together. So I think, yes, the neuropathologist will not do what Peter Berger did, the look under the microscope and just tell us yes, no, it is, this group or that group. But I think that there's even more of a specialist, whatever, the right term is, but coming through pathology to help us pull all this information together, some of which comes from computers and some of which, from the gross specimens. So I think their job is secure, as is ours in taking out these tumors for the moment, although it'd be nice to have better ways of doing that as well. As far as equity, that's a difficult problem. I think that it's, I feel on the one hand, it's our obligation in academic medical centers and in big private groups that are able to do clinical studies to pull the best technology together and to advance the field. Once the field is advanced, you would hope that systems become easily available in many different centers. One example I can think of that is that, one of the people here was with the original company when we did the first studies on navigation. It was only done in 3 centers. It was at the Montreal Neurological Institute, at Hopkins, and at the Barrow. And it took an hour and a half, 2 hours of operating time to put the fiducials right, it set up the computers. And, I remember my chairman at the time when I was just starting said, real surgeons don't need computers to find tumors, just like do the damn job and get out of the room. But where is navigation today? I don't think there's an operating room anywhere, that does modern neurosurgery that doesn't have a navigation system. It's become very equitable, if you will, for neurosurgical centers. I also think, Jim, that the issue is even more complex, because the question is whether patients with brain tumors should be funneled as a society, whether they should be funneled into major centers. I'm not going to say academic medical centers, but major centers, tertiary or quaternary centers, because the care is very sophisticated, and we do deprive people of the access to a lot of the technology, even just the operative technology, intraoperative MRIs, navigation systems, all of that. That's not available every place, and that maybe we're places in underserved areas, they're only doing a handful of these operations. They should be sent into centers that can do that. So I think that's a society issue. It sounds self-serving when we advocate for that, but I think as a patient advocate and equity issue, it really is the right thing to do.
The question: Are you getting intraop tumor genomic information? Like, is it IDH mutated and hence grade 4 astrocytoma, which is new CNS 5th edition, or if it's hypermethylated, maybe you can pull back your resection and they'll respond better to radiation and TMC, et cetera. Or that's the next few years. I think you did allude upon that earlier.
Yeah, it's a simple answer: no. We wish we did, and we're waiting an inordinate amount of time. It's incredibly challenging that for many patients, we have the histology very quickly, and then we're just, they know they have a malignancy, and we just say we have to wait, and weeks sometimes go by before we get all the information, and that is a incredible flaw in the way we're currently doing things. So, no, we have not figured out how to do that rapidly, despite the resources that we have.
Excellent. Thank you.